Nancy, Arthur, Sean, Luke, Ryan, Melanie, Rita, okay. So, Steve does go.
No, it's done now, yes.
Okay. That's what I was thinking, but just in case. So, okay, first of all, before I forget, next week we are having our first practical activity on Thursday, but on Wednesday, next Wednesday, we are having the next reinforcement activity, right? Okay, so, I'm not sure we will finish today with Unit 3, depends on how fast it will go, I don't know. So, I'm just thinking that, because I have another group that with them I'm going to do this reinforcement activity about Unit 3, but with you, as I haven't finished it yet, I'm thinking about doing the reinforcement activity about Unit 4 that we have already finished. Yes, Sam?
We have a lesson on Wednesday.
What?
Wednesday.
On Wednesday, it's normal class.
No.
Normal class.
Thursday. We have a Monday and Tuesday. Sure. We see you twice a week. Monday and Thursday.
Please, can anyone explain to me why we don't have class on
That's normal.
and just
Because you can't
next Monday?
We're N13.
Okay, no Monday. Okay.
Tuesday, Thursday.
The one we have. Yes. I know. We actually have classes on Monday and Thursday, and on Thursday we have the practical activity.
C'est jeudi.
Long day, guys. Long day.
La semaine prochaine, après la reinforcement.
Okay, so a solution is that in 2 weeks we are doing 2 reinforcement activities. So tell me when we are going to do them. That's Wednesday.
Teacher, wait. Teacher, just next Thursday one. Next Thursday one.
Into Thursday.
Non,
Thursday one.
No.
And the next Monday after the Thursday.
Yeah. On October 24th. You say.
Yes.
Comme ça, on va pas se poser.
What? ¿Y qué hago, Sean? ¿Y qué hago? ¿Qué hago? A ver. Vamos a ver.
That's the practice.
Let me think.
But she's saying that that's the two.
It's really too soon if we do the reinforcement activity next Monday, right? I want to try. So, okay, guys, listen, please. Okay, so we are doing the reinforcement activity about Unit 3 on October 21st, Monday, October 21st, yes? And the reinforcement activity about Unit 4 that Thursday, that week, the 24th. Is it okay?
But why we don't do? Why we don't change? Because we're just going to start doing 3 now, and we actually
But
finish doing 4.
it's so easy, Rasim. I mean, this is just one presentation.
I hope.
I guarantee that this is much easier than Unit 4 and it's easier to study. So, this is just a proposal. If you want, we can do the reinforcement activity about Unit 4 on October 21st, and that Thursday we are doing the activity about Unit 3. What do you prefer? So, first option, which is first reinforcement activity of Unit 4 and then Unit 3, or the other way around. Unit option 1. Raise your hand.
Option 1 is doing Unit 4 first.
Do you prefer first Unit 4 and then Unit 3, or the other way around? Unit 4 first. And the other way around?
Donc on aura une 3 en tête.
I think you win. So Unit 3 first. But guys, take this into account. You are going to do next week the practical activity about a skin. So you will have more fresh those contents to do the reinforcement activity next Monday. Yes? Yes? So, I will write it down just in case anybody is lost. Okay. So,
Alors vas-y, les gars, c'est que vous pouvez tout ça. On s'en bat les couilles.
I will write down.
And can you have the date of the midterm because
Yes, that's another thing I wanted to tell you. Okay, so everybody agrees with this? Yes? Perfect. So, Kimi, write it down.
Thank you.
Okay, anyway, midterm. This will be, or better said, I'm sure with a at 90% that the midterm will be on November
5, 8,
7.
5, 5, 8, 5, 5.
So, guys, okay, why this? The problem has been that, as I explained to you at the very beginning, we wanted to do a final exam. So that was our proposal, but it wasn't accepted. So to just search for spaces, classrooms to all of you, because this subject has more than or almost 500 students. So it's very difficult to look for all those spaces, okay? Because in those weeks you are having more midterms, so those spaces are already booked or occupied, yes? So the only day available at any time was this one to do the midterm, okay? I'm very sorry because I didn't want you to have this midterm earlier than you thought at first. But I'm going to try to do a lesson previous to the exam to review everything that we have studied, okay, in case you have any questions and all of that. Yes,
The
Aaron?
midterm is going to be Unit 1 to Unit 5?
From Unit 1 to Unit 5.
5.
Included, yes.
And then the final will be 6 to...
Exactly. Remember that if you pass this midterm, then for the final, you just have to study from Unit 6 to Unit 9. In case you fail, everything. Okay? Just for you to know. Okay? More questions? No?
Did you ask about the digital box?
Yes, I've asked, but we need to agree when to open them because there are a lot of students of late corporation that have similar problems. So the sooner I know when to open the digital test 1 or 2 in case you have missed them, I will let you know. Yes? Okay, more questions about this. So, you are probably a little bit afraid or nervous about the midterm being so early. Okay, so guys, what I recommend you is that please try to study a little bit. Not every day because I know that you have other subjects and you have a life, okay? But please, the midterm is much easier than the final exam, so try to pass it. Yes, sir.
You know the exam? Are the questions like the reinforcement activity questions?
Okay, the exam is a multiple choice question test. And yes, there will be similar questions. So that's why I really encourage you to do the reinforcement activities. So that's a way to push you to study or to review daily everything that we see here in class. Okay? Yes.
You said if we pass this, then we only have to do units 6 to 9. Does that mean like we won't get questions from 1 to 5?
No, if you pass, you don't have to study again units from 1 to 5. Yes?
Say you fail, you have a different test to everyone else.
Of course.
Teacher, you mean by not studying? No. The final is going to be on unit 1 to 9.
Okay.
No. Elle est en train de dire que, like,
So,
you do not study
guys, imagine if you have a 4, for instance, in the midterm, then you have to study again from unit 1 to unit 5.
And also
from
5 to
also,
9.
Exactly. Everything.
The final is going to be the same subject for all.
What?
The final is going to be the same subject for everyone.
Yes.
Yeah?
I mean,
Yeah,
you have to study the same.
Yeah, of course. Okay.
But obviously the questions will be different.
It's just for core fraud, but we're going to have the same question and everything. Huh?
No. I mean, you have the same questions, but yes. Different. So, more questions. If you have any question about the theory that comes to you when studying, please let me know before the classes so that we can stay a little bit after or just ask for office hours. Okay? That's it. So, yes? Fine. So, let's start with unit 3. Okay, so what we are going to see today are the general characteristics of the tegumentary system, the structure of the skin, and if possible, if we have the time, we will see other structures associated with the skin. But if not, we will see next week and in the practical activity. So a lot of things will be very repetitive so that you have them in mind. First, a structure of tegumentary system. What is the meaning of tegumentary? It refers to tegumentum. That means convert. That's the case of the skin, which is the largest organ in the body. Apart from having the skin in which we are going to find different types of tissues, not only epithelial, but also connective, nervous tissue, and the muscular tissue is somehow associated, but it is not part of the skin. But we will find muscles under the hypodermis. So, these accessory structures also associated are the hair, the nails, and the glands. We are not going to study the nails, but the hair and the glands also, we are going to see them under the microscope on this first practical activity. So, which are the general functions of the tegumentary system? First of all, it is the first line of defense against any microorganism that wants to invade us or other external agents in the environment. It will also help us to regulate our body temperature, as we are going to explain later, and if there are any changes regarding external stimuli, no matter if they are physical, chemical, related to sensations, for example, to pain, whatever, tactile sensations, we will have structures that will notice these changes. Also, we are going to see that this skin will be able to secrete waste substances, for example, the sweat by the sweat glands. And finally, we have another important function, which is the production of vitamin D from the ultraviolet radiation. Okay? It's not only that we intake vitamin D by the diet, but also being exposed to the sunlight will make us to produce vitamin D. So, regarding these skin facts, I'm only interested that you know the pH, which is acid, compared, for example, to the pH of blood, which is a little bit basic. It's around 7.3, 7.4, but the the pH of the skin is acidic. In some parts of our body, we are going to see hair covering this skin to protect us too. But the only exceptions to that will be the palmar and plantar region of both hands and feet, right? And one important thing is that everyone will have a specific tone, skin tone, depending on how much melanin our skin produces, okay? So the more tan you are, the more melanin will be produced. But also the melanin will give the color to our hair and our eyes. And finally, the skin also is involved to homeostasis due to the fact that it is a protective barrier that wants to maintain the internal homeostasis against an invasion or anything that can be unbalanced, let's say. So, now we go with the general structure of the skin that we have previously mentioned, just in general, but we already know or slightly remember that the skin is made of two main layers, the epidermis and the dermis. Respectively, we find epithelial tissue in the epidermis and connective tissue. As the epidermis, made of epithelial tissue, is avascular, then we are going to find in the most superficial parts of the dermis all the blood vessels and nervous terminations, nervous endings, so that they will give the irrigation and innervation to the epithelial tissue that forms the epidermis. And finally, we have mentioned several times that one important part made of adipose tissue that is related to the skin but is not part of this skin is the hypodermis. So that's why it's also known as subcutaneous tissue, right? Apart from the adipose tissue, we also find areolar connective tissue within the hypodermis, and the general functions that we probably can think about are the energy storage and the physical protection. Okay? And the hypodermis is also going to help to the thermoregulation due to this isolation by the adipose tissue. Right. So, here, guys, you have two images that correspond to histological samples of the skin. That is what you are going to see next week in the practical activity. So, using an optical microscope, you will be able to see all these parts of the skin and distinguish not only these layers, but also other structures. So, in here, what we can see is mainly the epidermis, which is a very thin layer compared to the dermis. We have mentioned previously that the dermis is also divided into two parts. One which is more superficial and is next to the epidermis, and one which is the deepest and very close to this hypodermis, in which we are going to see different structures, for example, hair follicles, glands, and so on. We will see them, don't worry. So, important. In this superficial part of the dermis, what we are going to observe mainly sometimes, well, most of the times, if you see little holes, will be these blood vessels that will provide this irrigation to the epidermis. But in here, if you are asking yourselves, what are those things? These correspond to sweat glands, okay? To the ducts of sweat glands. We will be able to distinguish these from sebaceous glands, for example. And in here, I think it's one of the easiest tissues to be recognized. We find the hypodermis that is made of adipose tissue, okay? Here you have an example of how a blood vessel looks like. If you see a very, very small or average hole in which you can find inside little red dots, those are red blood cells. So they correspond to a blood vessel. In between, we are going to see the composition of a loose connective tissue, areolar connective tissue. These are sweat glands, but don't worry by now, we will see them. Okay, so first of all, we are going to explain the epidermis, okay? So the most important facts that you should know for sure are these ones. First of all, we need to know the type of epithelium that is covering the skin, which is keratinized, stratified squamous epithelium. Always keratinized and stratified, okay? As we have mentioned, the epidermis is avascular. In the most deepest layer, which is the stratum basale, we are going to find different nervous sensory ends. But Sarah, you have said that all those sensory ends are in the most superficial part of the dermis. Yes, I have. But those nervous endings will be connected to particular cells found within this stratum basale that will send different stimuli to those nerve endings. We will see them, don't worry. So, which or how many cells, important cells, are we going to see within this epidermis? 4 types that we're going to explain right now, which are very, very tightly bound among them. And as you can observe in here, the epidermis will be divided into 5 strata or layers. From the deepest one to the most superficial one, we find the stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, and stratum corneum. Okay? Right. So first of all, let's start with the cells. As you can see, these are how they look like. these four main cells. So the most abundant one, as you can observe, are keratinocytes. Obviously, they will have keratin or produce keratin. Second, we find the melanocytes, the responsible ones of generating melanin. Then we have the Langerhans cells that are going to be responsible of the defense against microorganisms, and the Merkel cells. That's what I was saying before, guys. So here you observe a cell, right? This is the Merkel cell. But these cells are associated with a sensory neuron, with a sensory ending. This sensory ending of a sensory neuron is what we know as tactile disc or Merkel disc. So, all this together is known as Merkel cell. So, these cells will be found in the stratum basale, in the epidermis, and this sensory neuron will be just underneath in contact, but still in the superficial epidermis, okay? In the dermis. Right. So, characteristics of, yes,
you say that the Merkel cell, we find them in the stratum basale.
Exactly. But these sensory neurons, these endings, which are known as Merkel discs, are not in the epidermis, but just underneath in the dermis, okay? Fine. So, characteristics of keratinocytes. We know already that they are the most abundant cells in the skin, but we are going to find them also arranged in every single layer that we have already mentioned, okay? From the deepest one to the most superficial, we are going to see different development stages as the keratinocytes will be created in the stratum basale, but as they go up until the stratum corneum, they will degenerate and die. So their morphology will also change. So they will be in continuous renewal, okay? They receive this name because they produce keratin. What we're going to find within their cytoplasm are intermediate filaments that are known as tonofilaments, which I don't know why you don't have the name here, but I tell you. So these intermediate filaments are known as tonofilaments. These are important to be the scaffold of the cytoskeleton of these cells, the keratinocytes, okay? And they are also made of keratin, right? So, due to this important protein, the keratin, which is fibrous, they will protect us from different agents, from temperature changes, from microorganisms, abrasion, different physical agents, okay? And also within their cytoplasm, we are going to find what are known as lamellar granules. These lamellar granules contain a specific substance which is hydrophobic. So that's why these lamellar granules have this important function of water repellent. They will be waterproof, okay? To avoid that water penetrates to the skin and to avoid this invasion of microorganisms. Then we go with melanocytes, which are very related to keratinocytes, and we will see why. These are the second most abundant cell type within the skin, and they are mainly found in the stratum basale, okay? Obviously, they will produce melanin, but this melanin is contained in a specific granules known as melanosomes. Melanosomes. So, why are then the melanocytes related to keratinocytes? Because these melanocytes have a specific morphology. As you can observe with this picture, all these cells, these brown cells, have like cytoplasmatic projections that are known as dendrites. These dendrites are in charge of connect melanocytes to keratinocytes. What for? To pass all these melanosomes full of melanin so that these keratinocytes can also be protected against the UV radiation. Why? Because the main function of melanin when produced by the melanocytes is to make like a protective layer in our skin so that all the cells, all the structures can be protected against this physical radiation due to sunlight exposure. That's why when you are exposed to the sunlight for a very long time, you are more tanned, right? So this is the result of a lot of melanocytes working and producing melanin. So all this melanin will be distributed not only among melanocytes but also to other keratinocytes. So this melanin will go from the deepest layer to the most superficial one. So that's why we look more tan. Okay? Yes.
So then the projections of the melanocytes are touching the keratinocytes and it's putting in the melanin granules.
Exactly. All these melanosomes are the things that the melanocytes will pass to the keratinocytes. Yes?
No, I don't get it. The melanin is under keratin right now.
Wait. Here is more clear. Okay? So this is a melanocyte, right? The melanocytes are going to produce melanosomes full of melanin. Due to these dendrites, the melanocytes will be in touch with keratinocytes. So when, for example, we are for a very long time exposed to the sun, then the keratinocytes will be also protected against this radiation, thanks to having the content of the melanosomes, okay, full of melanin. Yes? Obviously, well, not obviously, sorry, but the keratinocytes still secrete keratin apart from melanin, okay? Okay, so that's it. Here you have the description of what I have just said. Questions?
It was about the melanocytes that absorb the UV are in the dermis, and after they.
They're not in the dermis. We are still talking about epidermis.
So the previous slide was all epidermis.
Everything that we are talking about is about epidermis.
So this is okay.
Okay. Only epidermis. Okay. So by now we have just explained the two types of cells found in the epidermis: keratinocytes and melanocytes. That's it.
The keratinocytes don't secrete the granules, right?
No.
It's the ones under the secretin.
You mean the lamellar granules? Or what do you mean? No. The keratinocytes don't secrete the melanosomes. No, they just receive them from melanocytes.
Who secretes melanosome?
Huh?
Who secretes melanosome?
Melanocytes.
Okay.
Okay? Right. Third type of cells, Langerhans cells. They are the third most abundant cell type, and they are mainly located in the spinosum stratum or stratum spinosum. Okay, so these Langerhans cells are very important due to their morphology too, because they are going to present also these projections, these prolongations, which are known also as not dendrites, but they also receive this other name, which is epidermal dendritic cells. So, due to this, their main function will be protecting us from the invasion of pathogens. So, they are going to generate the immune response within the skin, because they are going to present to our immune system the antigens that they recognize as strange. Okay? So, they are also known as antigen-presenting cells. As they are part of this immune system response, these Langerhans cells also derive from the bone marrow as other forming elements of blood. Okay? Right. And last type of cells, the Merkel cells or Merkel disc. So these Merkel cells are sensory cells located, as we have said, in the stratum basale and associated with sensory neurons, as we have mentioned. So their main purpose is just to recognize, to discriminate tactile sensations. So they are related to fine touch, to perceive if any tactile stimulus is received or we are noticing them. For example, if someone touches us, if something hits us, or anything related with touch. Okay? Right. That regarding the cells, let's see the composition of the different strata or layers that we have mentioned at the beginning. So, here you see this 4 or 5 different strata. But I have said at the beginning that there are 5. So why is that? Because one of these 5 layers will be absent in a specific parts of the body, which are the parts of our hands and the feet, which haven't hair. Those are the exceptions in which we are not going to find any hair covering the palmar region of feet and hands. So the stratum lucidum won't appear in these regions. That's why in this image you don't see any stratum lucidum. But first, let's start by the deepest one. It is the stratum basale. This will be on top of the basement membrane, anchored to it, and is made of an only single layer of cuboidal cells. So this is just, this is the only region in which we found this stratum basale, one single layer of cuboidal cells. So, what else do we find in here? Keratinocytes that will be produced by the germinative cells found in the stratum basale. So, these keratinocytes, as I've mentioned, will present a cytoskeleton made of tonal filaments. Also, we are going to find Merkel cells, some Langerhans cells, because they are mainly found in the stratum spinosum, and melanocytes that will be also dividing, so that melanocytes are mainly found in here, but will be also replaced as the skin is being renewed. So the main function of this stratum basale will be of proliferation, that is generating all these cells that form the epidermis. So we find mainly stem cells that will have a very high proliferation rate to differentiate into keratinocytes, melanocytes, or other of the two types of cells that we have mentioned. So apart from this germinative function, this stratum basale provides a structural support for the epidermis as it is based on the basement membrane.
What does germinative mean?
Germinative comes from germ, that is, it's the origin of the cells that will make the epidermis. Okay? Okay. So, second layer. Ernest?
Yes. I don't understand the link between the stratum basale and the keratinocyte. Can you just explain it?
The keratinocytes are found in the
Okay. In the stratum basale.
In the stratum basale, because as we find stem cells, these stem cells are precursor cells. They are going to differentiate into keratinocytes and or melanocytes, Merkel cells, as the
As
skin.
mesenchymal cells that we see before.
Similar.
Similar.
Exactly,
system.
similar. Exactly.
Okay.
I didn't really understand the present cytoskeleton.
So, in the cytoplasm of every cell, we need, well, they need a structure so that a lot of proteins and molecules need to be transported within the cytoplasm. That is what we know as cytoskeleton. This cytoskeleton is made by different types of filaments. One of these filaments is known or are known as intermediate filaments. In the case of keratinocytes, these intermediate filaments are known as tonofilaments. And these tonofilaments will be made of a molecule very similar to keratin, but it is the precursor of it. So, as keratinocytes are being generated, we find this internal structure within these cells so that keratin will be synthesized as these keratinocytes get or will be more mature. Yes?
I still don't understand the difference between germinative and proliferation.
The same. I mean, it's the same. Imagine, we have a stem cell. We all know what a stem cell is, more or less. Okay, so this stem cell, to differentiate into something, it needs first to replicate. Okay? So we are going to have more cells. Depending on different things, these cells that come from this original stem cell will be differentiated into one type of cell or another. So in the case of the stratum basale, what we find are these germinative layers or stem cells that are both the same. So when they replicate or they proliferate, they are both synonyms, they are going to give rise to different cells. Yes? Yeah? Got it? Now, second deepest layer, which is the stratum spinosum, also known as Malpighi's stratum. This is a little bit thicker because we find more layers, again, of keratinocytes. Remember that this cell is the most abundant in the epidermis and will be found in every stratum, in the 5 ones or 4 ones. So, why does this stratum receive this name, spinosum? Because under the optical microscope, when we have histological samples that are dehydrated, we are going to see that the cells are very, very close together by cell junctions, that look like thorns. Okay? So these things that you can see here, okay? These thin lines, they correspond to the cell junctions that we are going to find mainly between keratinocytes in the stratum spinosum. Due to their appearance, this stratum receives this name, spinosum, because these cell junctions, which are desmosomes mainly, they resemble to thorns. You know what a thorn is? Yes, everyone? Or no? No. Okay, imagine we have a rose, okay? You have a rose, and every rose has thorns. Yes, a thorn. She's a sticky. Not sticky, but yes, exactly. Okay? We all know now what is a thorn.
Yes.
Perfect. So, that's it. Related to the name. Why it's called spinosum? It's due to the cell junctions between the keratinocytes. But we find other things. For example, Langerhans cells, okay, and that will be projecting their prolongations just in case there is any strange microorganism to detect their antigens, okay, and to present them to the immune system cells, right? And also melanocytes, but in less number than in the stratum basale, okay? Also, these melanocytes will pass the melanin to the keratinocytes. So, the main function of this stratum will be providing strength and flexibility to the skin due to these tight cell junctions between keratinocytes mainly, which are the desmosomes, okay? Third, the stratum granulosum, very intuitive, because the keratinocytes, again, which appear in this stratum, will contain laminar granules that we have already mentioned, that they contain lipids, hydrophobic substances to repel water, so that it will make the skin somehow waterproof. Also, these cells will contain another type of granules, which they look darker, that receive this name, keratohyalin granules. Okay? So what is keratohyalin? This is a protein that will assemble the tonal filaments into keratin. Remember that at first, when explaining the stratum basale, we mentioned that these tonal filaments are the precursor of keratin, the definitive keratin. So, when we are in a more superficial stratum as the granulosum, then thanks to the action of keratohyalin granules, thanks to this protein, these tonal filaments will give rise to the definitive form of keratin. Okay? So, what happens little by little to these keratinocytes is that they will be full of keratin, but they will degenerate as they go up to the stratum corneum because they have a very short life. All the time the skin is being, it suffers from renewal. Okay, so more or less from the time a keratinocyte is produced in the stratum basale until the moment it is degenerated, more or less it passes like 4 weeks pass until this happens. So every month we are a new person because all the time the skin is being renewed. Okay? More or less, approximately. So the main function of this stratum granulosum will be preventing water loss, 2, and it will function as a physical barrier to microorganisms due to the action of the lamellar granules. Stratum lucidum, be careful here, only present in thick skin, only, because if we are talking about the rest of our skin, then it won't be present. What do we mean by thick skin? The palmar region of hands and feet. Those are thick skin.
Because there is no hair on it.
Because there is no hair. Right. So, stratum lucidum made of approximately from 3 to 5 or more layers of already dead keratinocytes. So their morphology now changes. Now they look more clear, flat, okay? And they won't have nucleus anymore, okay? As they are dead or degenerated. So, even though they start to this process of degeneration until they are just removed, these keratinocytes will contain large amounts of keratin. So let's try to connect these to something similar that we have already seen. Remember when we talked about the epiphyseal plates, when talking about the growth in length of bones? Remember these 4 zones in which the chondroblast will organize into columns and little by little they get calcified? Okay, so these chondroblasts, they will suffer from hypertrophy because they are full of matrix. Well, something similar happens to keratinocytes as they are in these superficial layers, like here, okay? So they will be full of keratin, but their morphology will change, okay? So this keratin has an important function of protection. We don't find keratin just in the skin, but also in our hair, right? Protecting it. Yes?
And where is the tona filament? Because where is the tona filament?
The tona filaments are the precursors, are filaments that will generate the definitive form of keratin, but we just have now the matter form.
Yeah, okay.
So, no tona filaments. Okay? Okay. You asked a lot today, very good. You are focused. I like it. So, last stratum, stratum corneum, the most superficial one. So, this is very thick compared to others that we have just explained. It is made of approximately 30 layers of flat dead keratinocytes, dead with keratin and surrounded by lipids. Why lipids? Because these lipids come from the the lamellar granules. Okay. So this stratum corneum will be a barrier to physical, chemical agents, different stimuli, and so on. Okay. So these lamellar granules will produce this waterproof action thanks to this substance which is water repellent. So what happens to the stratum corneum, as it is the most superficial layer, is that it will suffer from abrasion, friction, and so on. So it can change. It could be more thick so that a callus can be generated. For example, when you are when you go to the gym and you use a lot of different machines, then a lot of calluses are formed. A lot of guys are looking at their hands right now and oh yeah. Okay? Yes, calluses are formed in the hands. For example, in your stratum corneum. And this is a thickening of the epidermis. Okay? So, main function then of the stratum corneum being impermeable or waterproof and also protects the rest of the strata. So, a review of this. We have said that there are 5 strata in the case of thick skin, and just 4 in thin skin. So, the only stratum that is not present in thin skin is stratum lucidum. Then, also in thick skin, we will find a thicker stratum corneum, but it's just because of the difference in width, just that. Apart from this fact, we know now why all these strata receive their name and the different cells that we can find within them. Okay? So, if you are lucky, you will see these strata in the epidermis of the samples that you will get to see next week, okay, in the practical activity. Fine.
Well, let's switch layers of the hair. Which is the layer with the hair?
The dermis.
So you said that the soles, the palms and the soles are the only ones without hair, but does that mean it doesn't have a layer, it just doesn't have much hair?
They don't have hair.
Okay, but it's still the same number of layers. The only difference is it has one more because it's thicker.
No. I mean, so in the case of the palms of the hands and the feet, we are talking about thick skin. Thick skin doesn't have hairs. Okay? So that's why the stratum lucidum is present in thick skin, but not in thin skin. That's it. Okay? Yes.
I don't understand the difference between the stratum lucidum and the stratum corneum. Is that just the adipocyte cells? The difference between
No, there are no adipocytes.
No,
You mean the lipids?
the lipids. Sorry. Yeah.
Yes, that's.
Is this the difference? The main
This is
difference?
one of the main difference, and also the number of layers of
Okay.
dead keratinocytes. Yes, mainly the number of layers, exactly. As it doesn't appear in all the locations of the skin,
Yes.
that's one big difference. Fine. So, to finish with the epidermis, yes,
I
Aaron?
just have a question. So in the stratum corneum,
Yes.
we find the stratum lucidum.
No.
I
They
don't
are
understand.
different strata.
It's under.
Okay.
It is under, exactly. So, if we were seeing under the microscope a sample of the palm of our hands, we would see 5 strata, from the most superficial to the deepest one. Corneum, lucidum. What's next?
Stratum
Granulosum,
spinosum.
spinosum, and basale. That's it. Okay, very good. So, we're going to explain now how this growth and keratinization occurs, but we have already mentioned. So, what I've said is that from the moment a keratinocyte, for example, is generated from the stratum basale until the moment it is already dead and it is removed, this time is around 1 month. So, at the end, all these cells, these keratinocytes, melanocytes, etc., suffer from a constant cell division and growth or renewal. The thing about keratinization is that this process is only suffered by keratinocytes, of course, because this consists of the replacement of the intracellular content of these keratinocytes by the definitive or mature form of keratin, as we have mentioned. First, we have the tonofilaments that are the precursors of the final keratin. little by little, as they go up, they will be full of this keratin, and their morphology will change. They will get more flat, they will die, okay? So as they migrate to the surface, all this process will occur. We have new cells, they start migrating to the surface. The degeneration process that starts more or less in the stratum granulosum, and then when we are already in the stratum corneum, these keratinocytes are dead cells, okay? So, if for example, we have these abrasion or friction, or we suffer from them, or we suffer from any injury, we fall down, we have any cut, for example, injury, things, whatever, then the epithelial tissue is damaged, right? We need to repair it. So apart from the normal keratinization process and normal growth that happens every time in our skin, then we need to have a higher proliferation rate in the stratum basale, so that all these stem cells that we find within this stratum will start dividing even more due to the action of different growth factors, for example, the epidermal growth factor. When we are talking about factors in general, those refer to proteins that are going to enhance the activity of specific cells to do something. In the case of the epidermis, this epidermal growth factor will enhance the activity of the stem cells in the stratum basale, so that they can generate more cells. Okay, and that's it. So that's it about the epidermis. Now let's go to see the dermis. Okay, this dermis will be quite different from the epidermis. First of all, because the tissue changes. Now we are not talking about epithelial tissue, but about connective tissue, specifically areolar connective tissue. So, which cells are going to appear here? Fibroblasts, of course, as we are talking about connective tissue, also macrophages, and adipose cells if we are very close to the hypodermis. Okay? We are not going to find adipocytes all over the dermis. Be careful. The second main component of the connective tissue is the extracellular matrix. So, within it, we are going to find collagen and elastic fibers mainly that are going to provide the resistance and elasticity to our skin. Also, regarding the ground substance, the main component are those GAGs or glycosaminoglycans, such as the hyaluronic acid. That will be one of the most important ones to maintain the skin moisturized, okay? Among other things. So, then the main function of the dermis in general will be providing a structure so that the epidermis can be over something, which is the dermis. Immunological, thanks to the action of the macrophages. It's going to provide also mechanical protection. It is going to participate in the thermoregulation and water retention. We will see why it is involved in the thermoregulation. And something that we have already mentioned at the beginning is that in the most superficial part of the dermis is where we are going to find blood vessels and nerve endings. Okay? In the deepest regions is where normally hair follicles, glands, etc. can be found. Okay? Mainly there. So, we can say that this dermis is divided into two regions. So, let's name them and then we will explain the composition. You have them here. The most superficial region and the thinner, and which is thinner than the other one, is the papillary region. Approximately just this part in this drawing will correspond to the papillary dermis, okay? But the rest of it, the deepest portion, corresponds to the reticular region. So, in the reticular region, which is closer to the hypodermis, is where we see this hair follicle, this sweat gland, another sweat gland. We could also find sebaceous glands, etc. We will see the composition, don't worry. But first of all, let's talk about something else that we can find in the most superficial part of the dermis, the papillary region. We have mentioned that apart from the blood vessels, we find nerve endings. So, these nerve endings will correspond here in the dermis to receptors to detect a stimuli, different types of stimuli. But the thing is that we are going to find different types of receptors within the dermis, which are the ones that you find here. Mechanoreceptors related to tactile sensations, thermoreceptors, I guess you know what they detect, nociceptors, which are pain receptors, noci refers to pain, and proprioceptors. These proprioceptors are receptors mainly located in joints, in tendons, ligaments, muscles in general. And why they are proprioceptors? Proprio is own. Because these receptors will detect changes in our body, in our posture. They will also detect changes in the degree of stretching of our muscle, tendons, ligaments, and so on. So that's why they are mainly found within the muscles. Okay, so that if we do several movements, these receptors will send signals to our central nervous system to let's say integrate all those changes in our body posture, the movement, and so on. So the most important ones talking about skin will be these three ones: mechano, thermo, and nociceptors. Let's first talk about the mechanoreceptors, okay? Because they will be very important discriminating these tactile sensations, and we find 4 subtypes that you have in the slides. These 4 types are Merkel discs, Meissner corpuscles, Pacinian corpuscles, and Ruffini endings or corpuscles too. Yes?
The Merkel disc is the only one that is connected to the Merkel cells in the epidermis.
Correct.
Okay.
Exactly.
And the rest of the connective detecting sensation are not connected to epidermis cell.
No?
Okay.
That's a good difference. Okay. So maybe someone has thought, okay, why 3 of them has this surname of corpuscles, but the Merkel discs don't? Because these nerve endings will be covered by a special structure made of myelin that are those corpuscles. Do you know what myelin is? Myelin sheath, nerves, you know more or less, approximately, or no?
No.
Never mind, we will see what myelin is when we talk in Unit 6 about nervous tissue. Okay, myelin sheath is basically the cover of some nerves so that the speed of conduction of these action potentials or just the nerve impulse is upgraded. I mean, gets faster. But it is also something very important to other processes that we will see later. Never mind. Yes.
So only those you have in mind.
The corpuscle. The corpuscle.
I mean,
In here, I don't have this slide. I want it just in the Spanish group. Never mind. Yes.
So we find all these receptors only in the papillary region.
Wait. Wait. I haven't finished. Okay. So I repeat, guys. These three only have corpuscles, which are structures made by an extra cover of myelin. But don't worry about what a corpuscle is right now. It's just to know how they look like compared to a Merkel disc. As you can see, these Merkel discs are nervous-free endings. They are not covered by any structure, any corpuscle. So, where do we locate these four mechanoreceptors? Two of them, which are the Merkel discs and the Meissner corpuscles, will be found mainly in the papillary region of the dermis. That is, the most superficial part of the dermis. In here, we see this Merkel disc. We can also find these little corpuscles that correspond to the Meissner ones, which are very, very close to the epidermis. So we find them in the papillary region of the dermis. So what about the other two? Both Pacinian and Ruffini corpuscles will be larger structures, larger corpuscles, and will be found mainly in the reticular dermis. Okay? So there are also differences regarding what type of stimuli or sensations they detect. About these two, Merkel and Meissner corpuscles, they are related to fine touch. They are related to detect, in general, tactile sensations, also stretching of the skin. But they could also detect for how long a stimulus is detected in the skin. For example, if someone touches me for 1 second, or if the stimulus is more prolonged, okay? These two will also notice for how long the stimulus acts. But this is not the case, or not so much, of the Pacinian and Ruffini corpuscles. Why? Because these two are mainly in charge of detecting vibration, pressure, stretching sensations. So, in the case of Pacinian corpuscles, they will detect mainly pressure. So, if someone pushes me, these Pacinian corpuscles will say, okay, a specific stimulus. So, that these are the ones to detect this type of sensations. As in the case of Ruffini's endings, but they can also detect vibration and so on, okay? And what else? More things. So, we have now talked about the Nickel receptors, 4 types. Let's go with thermoreceptors. Sometimes these thermoreceptors can be also free nerve endings. In this drawing, they don't appear, but they can be divided into receptors that detect heat and others that detect cold. So that's, for example, if a stimulus or any object is very hot, specific receptors will be active when the temperature is higher than, for example, 30 degrees. If this temperature reaches more than 40 or 45 degrees, then the nociceptors starts to be active. Okay? That is the case of, for example, if we are very cold and we don't have gloves, our hands start to be numbed. Okay? And sometimes we even feel pain. That is the reason, or that's the consequence of the cold receptors being active under specific temperatures, for example, under 5 degrees. Okay? At those temperatures, we start to feel pain. Right. Nociceptors. They are also free nerve endings. And these nociceptors will detect any painful sensations just to keep us away from something that is causing us that pain, not just to be maintained or just to keep this stimulus or painful stimulus. Okay? In general, those free nerve endings, in here you have this nociceptor painted in this figure, in this image, but sometimes these free nerve endings can correspond to neurons that can detect other types of stimulus. For example, itching, tickles also. Yes, tickles, itching. I'm just thinking about the translation. Changes of temperature and pain in general. So, just to make a summary, free nerve endings can detect pain, itching, sensations, tickles, temperature changes, and other things. Okay?
Or that's the free nerves ending.
This type of
And
stimulant.
what about the mechanoreceptor also?
Yes, a nociceptor is specific for painful sensations. But nociceptors are free nerve endings.
But what is the main difference between the nociceptor and the mechanoreceptors?
Because mechanoreceptors are exclusive for tactile sensations. Okay? Nociceptors for pain.
So you're saying that if some of the nerves with the free nerve endings can be connected to other nerves that are corresponding to the itching or tingles and stuff?
It's complex because sometimes the stimulus is difficult to define. I mean, different sensations can be in between pain, itchy, something which is itchy, numb. When you are numb, it is not pain, it's what it is. So yes, different receptors can be active. It does happen.
So what does the free nerve ending do?
A free nerve ending is just a nerve that is not covered by distractors like a corpuscle.
And then we're saying that nociceptors, the itching, pain, they can follow.
Nociceptor is just a receptor that is a free nerve ending, but just detects pain. That's it. Okay? But there are other free nerve endings that are not nociceptors that can also detect those sensations. That's it. Okay?
In the previous presentation, all of that are all mechanoreceptors.
Yes.
You didn't give us
The cross and both. What?
no, you didn't give us an example of thermoreceptor or
Because they don't appear here.
okay.
But
In the superficial layer, it's only mechanoreceptor.
yes.
Yes,
Yes,
and
yes,
I
yes.
don't understand the thing of free.
Okay, guys. You see here nerves, right? Free nerve endings. But at first sight, you don't know if it is a nociceptor, if it is a nerve ending that detects itching, it detects temperature changes. So what I was saying is that these free nerve endings can be specialized in detecting pain, such as the case of nociceptors, or they can detect other things.
I know how to identify the free nerve ending.
You don't have to identify them. It's very difficult. Yes.
You said that free nerve ending are not covered by what?
by corpuscles. Corpuscles. Yes. 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 Okay. Okay, guys, I'm just going to explain to finish the lesson the dermis, and we go on next day with the accessory structures, okay? Few slides more.
You know the mechanoreceptors, are they only in the papillary region?
Two of them are
Which one?
the Merkel disc and the Meissner corpuscles. Right. So we have finished talking about the epidermis and its structure, the cells, and now we're going to focus on the structure of the dermis, on these two regions or layers that we have commented, starting by the papillary region. So first of all, this papillary region is made of areolar connective tissue, mainly, okay? So we're going to find collagen fibers and elastic fibers. This is the closest one to the epidermis, and sometimes we can find, I repeat, sometimes, pilus or hair follicles, sebaceous and swell glands, free nerve endings that can detect, as I've said, pain, warmth, coolness, tickling, and so on. Okay? And the fine touch receptors that are, tell me, which are the receptors, the mechanoreceptors that are located in the papillary region that we have just explained. And Meissner corpuscles. Good. Good. That's it. Okay? So, maybe when you have seen the images of the epidermis and the dermis, you have noticed that several structures that resembles some hills, like little mountains, appear in this papillary region. Those correspond to the dermal papillae. These dermal papillae are these regions of the papillary dermis are part of the dermis but serve as an increasing way, as a way to increase the surface of the skin. Okay? So, here you have. But when these dermal papillae are generated or are created, may I go on, please? 5 more minutes. So, when these dermal papillae appear, what happens is that the epidermis suffers from like an invagination to enter to the dermis. So, these structures are known as epidermal ridges, and they are these structures, the epidermal ridges. So, thanks to the epidermal ridges and the dermal papillae, the contact surface of the skin increases. Okay? So, in this dermal papillae, in the papillary region of the skin, at the end, is where we find the capillaries, the receptors that we have mentioned, and also these free nerve endings, mainly to provide to the epidermis with innervation and irrigation. So, now, reticular region, okay? This region is not made of irregular connective tissue, but we find now dense irregular connective tissue. So, what is more predominant in here are collagen fibers, very abundant. They are orientated to all directions that will provide resistance to the skin, and also elastic fibers that will give this ability to the skin to be elastic. So, as we have said, this reticular region, as it is the deepest one, is where we are going to find sweat glands, sebaceous glands, hair follicles, the Pacinian and Rufinis corpuscles, and of course, fibroblasts, as we are talking about connective tissue, but if we are very close to the hypodermis, adipocytes too. So the function will be provided elasticity, resistance, blah, 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 we already know. So, in here you have a very good drawing of everything that we have explained by now. Do you see? This region of the dermis will correspond to the papillary layer. In here, we will find this dermal papillae. Mainly in this dermal papillae is where we see these Meissner corpuscles. You see? We can also find some free nerve endings, as the one in here, that look like trees but without leaves.
Right.
And we can also distinguish this network of capillaries in the papillary layer. When we go down to the reticular layer, we see bigger corpuscles that correspond to the Pacinian and Rufinis corpuscles. And we see these pinkish lines correspond to the collagen and elastic fibers of the dense irregular connective tissue. And finally here, this adipose tissue is the hypodermis. Okay? Questions? Not at all. Okay. I leave you alone. You can go.
En réalité, le plus facile, c'est beaucoup d'infos, c'est c'est vraiment inutile pour dentaire. Attention à hein? Ah non, on n'a pas de non, moi j'ai pas de cours.